Welcome, everyone, to this plenary on conservation genomics. My name is John Kress. I'm Curator Emeritus at the Smithsonian Institution. My field is tropical ecology and evolution, with a great interest in how genomics is going to give us some answers to a lot of important questions. I'm also co-chair of the Earth Biogenome Project. It's my great pleasure to chair this session this morning on conservation genomics. All of us at this conference know and are aware that biodiversity around the world is in crisis mode at this point. We've heard that over and over again at the plenaries, from contributed papers, and even in poster sessions. To accurately track genetic degradation and species loss in today's world requires in-depth knowledge of the ecology, evolution, history of the species that occur in these degraded habitats. Genomic data will provide the raw information that we need for understanding how life evolved and how it exists today, and how we can help it exist in the future. This plenary address on conservation genomics will highlight the contribution of genomic data to assessing genetic diversity in both currently and potentially threatened species, and more importantly, developing strategies for species conservation as our habitats continue to degrade and disappear in the Anthropocene. I just wanted to remind everyone that this plenary talk by Dr. Carolyn Hogg on conservation genomics will be followed by Stream B, chaired by Rebecca Johnson, with 8 additional invited and contributed talks on the same subject. And moreover, there's a poster session this afternoon or later on in the conference, depending on where you are in the world, that will have an additional 12 papers on conservation genomics, including such topics as population structure, hybrid zones, genetic erosion, inbreeding, evolutionary relationships, mitochondrial genomes, and population divergence. So there's a lot that you'll hear about in terms of conservation genomics, not only in this plenary session. Now, to our plenary speaker, it's my great pleasure to introduce Carolyn Hogg. She was trained at the University of Western Australia, where she received a bachelor's degree in marine biology and zoology, went on to receive honors in the biological sciences at Murdoch University, and then received her PhD at the University of Sydney in veterinary sciences, where she currently is at present. She serves now as the Senior Research Manager of the Australian Wildlife Genomics Group, which is part of the Vice Chancellor's Research Office at the University of Sydney. Carolyn has numerous publications, both academic publications, but also dozens and dozens of industry reports, which are actually the kind of the work side of what she does as a conservation biologist. Perhaps most importantly is her recently published book that she edited with a number of other scientists, called Saving the Tasmanian Devil: Recovery Through Science-Based Management, published by CSIRO. She may mention that book during her talk. She told me that she has had an eclectic career working all over the world in foraging ecology, reproductive biology, captive breeding, translocations, and conservation genetics. She now calls herself a conservation biologist. And she has worked on all 7 continents, and most recently in Antarctica, both as an academic researcher and as a conservation manager. In May of this year, she helped to launch the Threatened Species Initiative, a collaborative project to develop genomic resources for Australia's threatened species. And I think this will be part of the topic again of her plenary address. If you read her abstract carefully, there's one statement in there that I think is probably most important. And I quote, "A reference genome does not conserve a species. Having the reference genome allows us to develop a suite of tools to understand both genome-wide and functional diversity within and between species. Conservation practitioners can then use these genetic resources to inform their decision-making." It's my great pleasure to introduce Carolyn Hogg. The title of her talk is The Fourth Industrial Revolution and the Age of Extinction. We will have questions afterwards. Carolyn.
On the 24th of December 1968, William Anders took this photo from the Apollo 8 mission, an image which has been declared as one of the most influential environmental photographs ever taken. But to me, it tells a story of one of technological marvel, of fragility and of hope, and of our biodiversity. In the 20th century, we saw the rise of the digital age as we developed greater and better technologies, which has led some to call the 21st century the fourth industrial revolution, characterised by the fusion of digital, biological and the physical worlds and our growing use of the new technologies. When Bill Anders took this photo, the internet, artificial intelligence, machine learning and genome sequencing were all simply things of science fiction. But 50 years later, all of these exist and the rate of technology is accelerating. Also, in that same 50-year period, we've lost 330 species have gone extinct. And with this, the rate of extinction is accelerating. You just now need to turn to any form of media and there is a monthly or even a weekly reminder of the current extinction crisis in which we find ourselves. Last year alone, the United Nations estimated that 1 million species are now at risk of extinction, and we are mostly to blame. If we look at the IUCN Red List, there's 13,500 animal species currently listed as conservation dependent or worse. And if we look at what we're here today to talk about, genomics or genome sequencing, less than 8% of these 13,500 species have a genome available to them. If we dive deeper into what we know is available at the NCBI, we find that we are starting to look at a wider variety of taxa across animal genomes, but still less than 6% of current or published genomes are conservation dependent. So this has led many to have this conversation about what we should be doing and how we should be developing genomes for conservation. We know that genome sequencing has come down dramatically, the costs in the last 10 to 15 year period. And in that same time period, we've seen a significant increase in whole genome sequences and other types of gene sequences available to us. At that same time period, we've had a very large amount of bioinformatics, large amounts of compute, as seen by the number of bioinformatic publications that have occurred at the same period of time. Unfortunately, what's happening now is we've got this massive increase in technology, in genome sequencing, in bioinformatics, and the compute technology required to compute large amounts of big data. And we're ending us up with a gap. On one side of the gap, we have the geneticists and genome biologists who are actively working to develop these genome libraries, to develop something that can record all forms of life that we have on the planet. On the other side of the gap, however, are the conservation managers like myself, ecologists, conservation practitioners, policymakers, all pulling their hair out about how they should be using this big data and these genome sequences in their decision making. How should they be using this information when they decide whether or not they're going to translocate some animals, or in their policy decision making in regards to species protection and habitat protection? We really need to figure out a way that by bridging this gap is key to lowering the extinction rates. And without it, we're going to struggle to make sure that the genomes that we are producing become useful to the people that need them. Managers know that genetic diversity is important. So they know that genetic diversity and inbreeding are inversely related. They know that they need more genetic diversity or to maintain genetic diversity in the landscape within populations to make sure they can maintain adaptive potential for future change. And they also know that they need this to maintain fitness of individuals to ensure their survival. So why then are we seeing not many genomes being used in the conservation genomic space? For us here in Australia, genomes in conservation have always kind of been considered traditionally as a nice to have. Funding is not limitless, particularly in the conservation space. And what we find is that conservation managers will make decisions and hard decisions about whether or not they're going to protect habitat, whether or not they're going to translocate their species, going to take out the feral invasive pest species that are affecting populations. And so the last thing that they're going to spend their money on tends to be the genomic sequencing. And so what's happening is we're ending up with this gap between the genomic resources that are being made and the conservation management actions that could actually use that information and allow them to answer some pretty key biological questions, as well as some pretty complex biological questions. So if we look at our 200 vertebrate recovery plans in Australia, About 80% of them have a genetic action within the recovery plan itself, and that genetic action is targeted towards recovery of that species. But when you go and assess how many of those 200 national vertebrate recovery plans are in fact using genetic data, less than 15% of them are using genetic data at this point in time. So what does this mean for genomics and management? So when we started on this journey about how do you incorporate conservation genomics into conservation management practice, having a reference genome is great, but you don't necessarily need a reference genome for your species at hand. You can use a cogeneric species. And once we've got that reference genome, we can then create some really key information around these species. We can start to do some functional gene characterization, particularly important for species that have disease or you moving, translocating individuals and wanting to promote immune gene diversity in presence of disease. This is commonly seen in the orange-bellied parrot, which you can see at the top of the screen, and also in the koalas. Reproductive genes, if you've got a species that has a problem breeding, behaviour genes maybe, or heat tolerance, which we find particularly important here in Australia with our ever warming climate. And then, of course, the more traditional methods that most of you know about for assessing genome-wide diversity. So those are the SNPs, and we use those SNPs for population differentiation, population genetic diversity, assessing things such as inbreeding effects, and even in some species now it is becoming quite economical to start using whole genome sequencing to look at your genome-wide diversity parameters. So who are we? Well, we are the Australasian Wildlife Genomics Group, headed up by Professor Kathy Belov. And our research group has been focusing on two main key areas in relation to genomics for about the last 10, 15 years. And that is the area of conservation genomics, as well as immunogenetics. And we've worked on quite a range of Australian marsupial species in particular, and have contributed to most of the genomes that you can see up here on the screen. And more recently this year, we've started sequencing the orange-bellied parrot genome, the numbat genome, the woylie genome, and also the bilby genome. But it's not just enough for me to sit here today and to tell you that we need to make it easier for conservation managers to start to use genomic data. What do I actually mean by that? So what I thought I would do today is walk you through a couple of case studies and show you just how you can use the information by working in partnership with the conservation managers. So let's talk about this little guy, the Tasmanian devil. Tasmanian devils are the world's largest marsupial carnivore. Found only on the island state of Tasmania, off the south coast of Australia. They're about, females reach about 8 kilos and males will get up to about 10 or 12 kilos. They're about the size of a small dog. Unfortunately, however, Tasmanian devils have very, very low genetic diversity and have had low genetic diversity for probably about the last 20,000 years. What started in 1996, however, is devil facial tumour disease. So devil facial tumour disease, or DFTD, is a clonal infectious cancer. And we first noted it up here in the northeast corner of Tasmania at a place called Wackalina Mount William, where a photographer took a photo of a devil with these large tumours on their face. Slowly but surely, over the next 10 to 15 years, we started to see the disease move southwards and westwards across the state of Tasmania. And now we only have disease-free areas up here in the northwest of the state and the southwest of the state. And for the rest of the state, populations have decreased by about up to 80%. Unfortunately, in 2012, a second form of devil facial tumour disease, in fact, occurred down here in the southeast corner. And so the species is now suffering from 2 infectious clonal cancers. So what do we do? So this is Kathy Belov and myself teamed up when I was working at the Zoo and Aquarium Association back in 2010, and Kathy was at the University of Sydney, and partnering with San Diego Zoo Global and the Save the Tasmanian Devil program. We wanted to see if there was a different way we could approach the problem. So what you find is in traditional conservation research, you might have your academics or your researchers come up with a research idea. They get some funding, they do their research, they analyse their results, depending on what you find the results say. You might go back and conduct some more research, you publish it. That publication may be read, if it's open access, by some conservation managers. The research may or may not be implemented. You might have a management outcome, which may lead to a conservation outcome. So from start to finish, that can take anywhere between 8 to 15 years. In a species That is being born and dying by the age of 3 with only a 1.5 to 2 year generation time, an 8 to 15 year period is simply too long to start to implement good conservation action. So we wanted to know if we could change this and develop what we call the tools and tech approach. So could we take the management questions that we have, feed them into the research ideas, leverage our management funding into our research funding, and then have direct consultation between the researchers and the management team to try and make sure that whatever the research results we're finding could be directly implemented back into our management decision making. So we could have faster conservation outcomes based on our genetic data, while the academics and the research students went ahead and published their research data. So what does this mean for devils? So this is the approach that we started back in 2012, and we've had some pretty fantastic results to date. When we look at using genomics in the management of devils, we find that, say the Tasmanian devil program have an insurance population, which I'll talk about in a little bit more detail in a minute. And we released the devils to an island, a disease-free island, and then the question came about of whether or not we could start to translocate devils back into all these different populations across the state of Tasmania. And the reason we wanted to start to translocate the devils back into the wild is that we knew that these animals populations had decreased by about 80%. They live in a highly fragmented landscape, and they were starting to succumb to other pressures from roadkill, from interactions with dogs, and also due to inbreeding effects that we're starting to see at some populations. So the questions that the Tasmanian devil program came and started to ask us was, well, if we're going to do translocations, which populations should we start with? Where should we be sourcing the devils from? What is the survival rates of the animals once we release them? Do they breed when we release them? And what we came to realise is that we could use genetic and genomic data to actually answer these questions. And today I'm going to talk to you about just 2 locations, one being down here in the island, Marar Island, off the southeast coast of Tasmania, where we released devils from the zoo population onto Marar Island in 2012, 2013, and then we translocated devils from that island up to Stony Head in 2016. However, let's take a step back. So as the disease was sweeping across the state of Tasmania, we started to go out and bring in founding animals for an insurance population, a captive insurance population. So we captured some devils up here in the north of Tasmania at a place called Narantapu National Park in 2012, and in the southern corner at South Hastings Cave in 2005. And then the rest of the remaining devils we brought into the insurance population came very much from the west coast of Tasmania over here in the Woolnortha area and all the way down to Strahan. However, what we then realised is that we didn't really have any genetic representation from many other locations in the state. And so then we worked really hard to try and develop a tool set so we could bring orphans into the population. And we could bring these orphans in from the rest of the state. And so the orphan program allows us to acquire devils from diseased mothers, and they come in and they go through a series of quarantine steps before they get fully incorporated into the insurance population. What we also know, though, when we look at these populations, is that there's very poor connectivity in the wild. So we have a very distinct geographic differentiation in the wild devils between the West Coast animals and the East Coast animals. And so what we wanted to know was, if we took these animals from these 2 different genetically differentiated populations and we released them onto an island, onto Marar Island, how would they breed and what would the genetic admixture look like? So when we released the devils onto Marar Island in 2012 and 2013, we had a large portion of animals from the West Coast population, and some animals from the East Coast population, and a few animals that had been in captivity from before the insurance population started. What we know, however, is that if you look here, the blue is the West Coast devils that went to Marar Island, and the red is the East Coast animals that we ended up on Marar Island. And as you can see, with the grey individuals are all the individuals that have ever been born on Marar Island. And so what we know is that these animals are a genetic mix of both East and West populations, and you can tell by the spread across the y-axis that these animals are more genetically diverse than the individuals that we put out on the island originally. So this is work that Elspeth McLennan did during her PhD. And what Elle was able to show is that when you look at every single individual that was ever born on the island across the x-axis, and on the left axis here, we have founder representation. So how much of that individual's heritage is pure bloodline from the West Coast. The pink is the East Coast, and the little light peach area you can see here is the captive animals from the old zoo population. If we look at standardized heterozygosity here on the right-hand side y-axis, you can see that there's animals that have got a mixture of East and West bloodline in them, have much more higher genetic diversity or standardized heterozygosity than the individuals who are simply only from the West Coast heritage themselves. However, the question then is, okay, they did genetically mix, but then is there a benefit to this mixing? So we then had some work done by Rowena Chong during her PhD. And when you look at the both neutral expected heterozygosity on the animals on Maria Island and at Wool North, and this is using 33 microsatellite neutral markers and MHC2 linked microsatellite markers, we can see that we have more genetic diversity on the Maria Island population than the disease-free population up in the northwest at Wool North in the northwest side of Tasmania. When you go ahead and look at the fecal egg count data, again, we see that there's a large proportion of uninfected animals on Maria Island compared to the infected versus uninfected on Wool North. And when we then look at what the number of fecal egg counts or in the grams per fecal matter, we see that the individuals with much more heterozygosity or a lower internal relatedness value for MHC2 linked microsats, these individuals have a lower fecal egg count than the individuals that are more homozygous. So what we're seeing is not only is there genetic admixture on the island itself, but there is in fact a fitness benefit to the individuals that are more genetically diverse. So the question then was, well, if we take the animals off the island and we move them to a wild site in Tasmania, what's the impact of taking that action? So this is some more work that Elle did during her PhD, and what we found is that if we look at the animals, all the animals that we have on Maria Island, you can see they're genetically different from the local incumbent population up at Stony Head. You also see that Stony Head individuals are very tightly clustered together, which is an indication that this population has much lower heterozygosity than we find at the Maria Island population. And we see that again when we look at the internal relatedness, which is simply a measure of homozygosity. The animals on Maria Island are more heterozygous than the animals that we see up at Stony Head. So the question then from the Devil program was, when we release the animals, do the released devils from Maria Island go ahead and breed with the Stony Head incumbent population? And do they produce these hybrid individuals? And what do these hybrid individuals in fact look like? So what Elspeth found is that when we did the release in 2016, the cohort that was born in the first year after the release, the 2017 juveniles, the pink individuals here are the Stony Head incumbent adults. The blue are the 17 Maria Island individuals we released from the island. And the turquoise light coloured blue you can see in the middle are the offspring produced in 2017. So when you've got the very light turquoise animals clustering over here with the Maria Island adults, those are obviously a female that mated with a male she knew rather than someone she didn't. And again, we find that as well, that a number of the incumbent animals only bred with animals that were already existing in the population. What we do know though from the display of individuals you can see here in the middle between the dark blue and the pink, the juveniles, that is where they've got a hybrid. These are the hybrid individuals. So they've got one parent from Maria Island and one parent from Stony Head. And what does that mean for the animals at the site itself? And this is work both Elspeth looked at and Wan Cheng, a postdoc within our group, is actually starting to look at more closely, particularly in relation to the MHC region, and Wan will be talking about that a little later on today. But what we do know is that if you are purebred, so if you have only got Maria Island to Maria Island parents or Stony Head to Stony Head parents, then you have more homozygosity than the individuals that are hybrid. So the hybrid individuals are exhibiting greater heterozygosity than we would expect. What does that mean though? It translates to about a 1.5 times increase in genome-wide genetic diversity for the hybrid individuals and a 4.5 times increase in functional genetic diversity. So we're currently working really hard at this site. We're taking blood samples, fecal egg counts, DNA, RNA-seq, and so watch this space because there's going to be some pretty exciting results coming out in the near future. So the next question that we had was, well, we knew it would work with devils. So devils had a lot of funding and research capacity initially in that program. And we wanted to know whether we could use all that information that we developed around genetic and genomic sequencing with devils and how we could make smarter translocation decisions. Could we translate that information away from devils into another species? So let me introduce you to the bilby. This is the lesser bilby. The lesser bilby is an arid marsupial that's was found predominantly in the central region of Australia, and this is in fact the greater bilby on the right-hand side here. And the greater bilby is the light-coloured grey, browny-coloured area you can see is the old range of greater bilbies. Unfortunately, though, the lesser bilby became extinct in the 1960s, around about the time that we were putting man into space and Bill Anders was taking his photo of the Earth. However, the greater bilby is still alive today, but is living in only about 20% of their original range, and they're very much up here in the arid zone. But this is a species that used to exist in both temperate Australia down the southern coast area here and through the arid zone through the central part of Australia. So for bilbies themselves, we've already had one species go extinct, and what's happening with the other species that is now severely depleted in their range. What we do know is that we've had bilbies in captivity since 1985, very similar problem to what we have in devils. They live in zoos, they're found in fenced enclosures, they're also found on islands. And the management questions from the conservation managers for this species was, how different or similar are all these populations that are interspersed across Australia? How should bilbies be managed moving forward into the future? How do we make it a metapopulation? How do the animals that we have behind fences in large 10,000 hectare fenced areas to keep the bilbies away from cats and foxes, how do these populations compare to the wild populations? And how are they uniquely adapted to live in both this arid and temperate climate? And if we've got individuals that are adapted to a temperate climate and we move them to an arid zone, are we going to have problems with survivability for those individuals moving forward into the future? So this project's been undertaken by Paris Brandes as part of her PhD project, and Kate Farquharson, who's one of our postdocs. And so we've been sequencing and assembling the bilby genome. It's completely assembled and annotated now, and we'll be releasing this genome in early 2021 once we've finished doing the final polishing steps. What we also have is we're looking at functional adaptations for this species. So we're actually comparing a number of resequenced genomes of individuals from both arid and temperate regions within Australia, and also comparing some lesser bilby whole genome sequence data to the greater bilby. So we can have a better understanding of the differences between these two species and how one was solely adapted to an arid environment and the other species, the greater bilby, was adapted to both a temperate and an arid climate. And the reason we're really interested in these types of functional adaptations is because Australia is a very warming climate. And as our climate begins to dry out, as the climate changes into the future, we need to have a keener and better understanding of how our species can potentially adapt to a drying environment. And this will help us make more informed decisions when it comes for protecting habitat, particular areas of refugia where we think animals might go into as the habitat starts to dry out into the future. More importantly though, at the moment we've been working pretty closely with the management teams, the Australian Wildlife Conservancy and Save the Bilby Fund, Taronga Zoo, the West Australian Government and the Australian Federal Government all have investments in large fence enclosures across Australia. And these, there's about individuals that they're trying to move between populations, they're trying to start new fenced enclosures. Just last week we released bilbies back into Sturt National Park in New South Wales. It's the first time that bilbies have actually been in that location for the past 100 years. And so we've been helping these management teams with the population genetic data over using about 3,500 SNPs. Pretty common activity these days. We've sampled over 300 individuals across 12 sites. And we've created these easy to interpret PCOA plots for the managers so they can see where their population sits relative to the other populations. And we've created these really easy to use FST tables. So essentially looking at the genetic differentiation. If a manager wants to make a decision about where they should potentially be sourcing some new individuals from, the moral of the story is don't mix the populations that are red, mix populations that are green. And if for logistic and operational regions, you can't mix a green population, you can mix a yellow population. It's not the best, but we can cope with that moving forward. So why is all of this so important? And it's particularly important for us in Australia because of what happened in the last summer. So the summer of 2019 to 2020, it was a bad year start for us, and it just seems to have got worse. But of course, we had the mega fires across Australia, and these fires were the largest fires that have ever been seen pretty much in modern history. It was a result of a very long extended drought for the last 4 or 5 years and a very, very hot summer, as you can see in the map here from Geosciences Australia, the hot spots that were found in January this year. These fires burnt so fast and so hot that in some areas it still just looks like the surface of the moon. When you look at the landscape, even 8 months later, there's very little recovery to the bush. Most unfortunately, however, 1 billion animals were lost during this event, and we had 23 million acres of land and habitat have been burnt. How are we supposed to recover from this? This is going to take some serious conservation management actions in order to try and revegetate as well as rehabituate this landscape. When you look at Australia, we have about 8,000 vertebrate species in this country, and most of them are endemic. So most of them are not found anywhere else in the world. And when you look at the areas, this is a recent paper that came out in Nature, Ecology and Evolution, you can see that if you look at the southern part of Australia, which was the study area that they undertook the study on, the red bits are all the habitat that was impacted by fire. And when you look at some of the critically endangered or endangered species included in this study, large portions of their habitat was in fact burnt. And so how are we going to make management decisions in this landscape moving forward? So what we've done is we've partnered with Bioplatforms Australia, who, and building on their work that they did with the Mammal Genomes Project and the Australian and Amphibian and Reptile Genomics Project and the Genomics for Australian Plants. All those 3 projects were pretty much established to do some phylogenetics and develop some genomes and also undertake some conservation genomics. So we've developed the Threatened Species Initiative, and the Threatened Species Initiative is a little bit different from these other 3. It's designed to develop genomic resources for some of Australia's most threatened species, both plants and animals. But not only that, it's also to develop some associated population genetic data. So we can provide a platform or a jumpstart conservation managers being able to use the genetic data for these populations. But most importantly, the big aspect of this program is it's really being driven by the conservation managers and their need. So they have provided us with a list of species that they would like to see done. There's about 286 species on the list at the moment, and it's currently growing. But more importantly, we're wanting to develop an applied conservation genomics portal online. So it's an online toolkit for conservation managers. It's a place that they can go, they can download the standard protocols in how to collect their samples, how to get them sequenced, how to analyze the data, interpret the data, and how they can use that information in their decision-making moving forward. It's really about bridging the gap between all the different genomics initiatives and conservation management actions. So how does it work? Well, the idea of the Threatened Species Initiative is that we can build on all the reference genomes that have been assembled and annotated not only through our initiative itself, but other bioplatforms projects within Australia and the global genome initiatives such as the Earth Biogenome Project and Vertebrate Genomes and Bird 10K and Bat 1K, because all these genomes are stored publicly available in the cloud for us to use. So the idea is that the end user goes out and collects their sample. For us, this is us doing Tasmanian devil work. We've actually started collecting the samples using digital tablets, so the metadata can be transferred directly to the lab teams. The samples are then extracted and sequenced by the commercial provider. The sequencing gets done, the sample, the data comes back, the end user is very excited, receives their end user, their sequence data, and then they can go on and use our online platform, which will help them undertake some common analysis. And so essentially you go into the portal, this is just a little bit of a mock-up of what we're planning on doing. You can load up your data, whether it be reference aligned or de novo assembly. You can select a genome from the TSI or any other data set. So if it's got an NCBI accession number, it then takes you to the next step where you can load up your FASTQ files as well as your barcodes. You can then just select your downstream analysis, whether it be population differentiation, genetic diversity, inbreeding, or parentage analysis, relatedness, whatever types of management questions. Because what we found when we talked to the conservation managers, their questions are quite common across the board. You can then have some advanced parameter settings, or you can just use the default settings. And then you simply type in your submission details, declare whether or not you want some extra catalogue or VCF files, and then you hit submit and you end up with your submission summary. So this is for the end user who really doesn't want to change anything too much. They simply get their data back from the sequencing company and they want to analyze it and get their output report so they can make management decisions. But the portal also has a compute tab where people who want to undertake their own analysis or be more extensive in the analysis they undertake, they can do that. And essentially what happens when you submit your data to the portal, it fires up a high performance computer in the cloud and it lets you run your analysis quite quickly in the cloud environment and then pulls it back down and provides you with your report. So you get a very friendly end user report, receives their data, easy to interpret format, and then it allows the conservation managers to then go on and simply undertake their translocations or make their management decisions. So you look at that and you think, oh, it's all very good in theory, but how does it actually work? So this is us collecting devil samples at the end of January every year. The data, as I said, is collected on a digital tablet. The information is sent to the lab along with the samples where they're extracted. We then run it through our online portal system. We send it back to the guys on the ground and they make their translocation decisions. So we're collecting the samples at the end of January and we're translocating the devils in June. So that is this period of 4.5 months, which at the moment is as fast as we can get it. And the longest time is in fact getting the sample sequenced. So we're going to be working on how we can speed up that process. So where we're at the moment is we've set up a pilot project for the Threatened Species Initiative. And these are all the species that we're working on. So we're trying to work out our workflows at the moment to make it really simple for end users and for conservation managers to use the information. Some species have a reference genome, others don't. Some have some genetic data, some have no genetic data. But all of these species on this list are part of an active conservation program at the moment. These are all species that have been flagged as species that require genetic data in order to make informed decisions. And so the pilot project has just commenced and will complete in early 2021, and then we'll roll it out for more species across the state. This is by no means a small program. There's a significant and large number of partners involved. Most importantly, though, we have all the state and federal government threatened species managers involved in this program, including all the not-for-profit organisations that have the fenced enclosures, the zoos and the museums, and we're keenly supported not only by the Australian Research Council, but also have developed pretty strong partnerships with Amazon Web Services and Intel and Ronin to give us access to high-performance computing. So what this means at the end of the day is that on one side we have the geneticists and the genome biologists developing these fancy genomes in the genome library. But my question to everyone is, there's no use in developing a gold standard, diamond standard, platinum standard chromosome length assembly if the guys on the other side of the divide, being the ecologists and the conservation managers and the policymakers, don't actually know how to use that information moving forward. And as we develop more and more sequencing technology and make more complicated bioinformatic pathways, it becomes more and more difficult for those end users to be able to access the information. And so this is really what the threatened species is about. We know that there's 1 million species that are now under threat of extinction, and conservation managers and policymakers really need to make decisions in real time. And genomics can inform many of the decisions that they actually have questions for. And so the TSI has been designed and built to bridge this gap by partnering on both sides with the genome biologists and the geneticists and the bioinformaticians, along with the conservation managers and the practitioners and policymakers. Because it's not just enough to build the library anymore, we really need to teach others how to use it, because this responsibility rests with all of us. And so when I ask my conservation managers, why do we need to use genetic data? What do you find is the use and value of having access to genomic data? And one of them once said to me that having genetic data available for management of a threatened species is the difference between flying blind or flying with a navigation system. So coming back to where I started, over 50 years ago, Bill Anders from Apollo 8 took this photograph of the technological advances that we've just simply excelled at in this massive amount in the last 50 years. But in that same period of time, we've had 330 species go extinct, and the extinction rate is ever increasing. We now know that we can use the outputs of the fourth industrial revolution. We can use the outputs of the technology that we've developed to help slow this age of extinction. And I can say that because we've seen it with species like the Tasmanian devils and the Bilbies. So I leave you to ponder these questions. How are all of you who are creating genomes making them useful for the conservationists who need them? How are your actions making an impact? And more importantly, what will our planet look like 50 years from now?
Carolyn, fantastic. I think we're going to have to export what's happening in Australia to the entire world, which would be great. A lot of questions are coming in. So actually, I want to start out by asking one that maybe is the most general. And you mentioned at the very end of your talk, with now 1 million species estimated to be threatened with extinction, doesn't something like the Earth Biogenome Project, which we want to generate genomes for all species on the planet, become even more critical and more important for the sorts of work that you're doing?
Yeah, no, definitely. That kind of production of genomes over a 10-year period will be fantastic for us. But one thing that we recognise quite clearly is that at the same time that we're producing all the genomes for life on Earth, we need to upskill the conservation managers and the people who are actually going to use the information in their decision making, because there's quite a significant gap between the genome being produced and managers being able to use that information. And the Threatened Species Initiative has, we've got a pretty big vision of getting to a point where managers can start to use the information to make decisions, but they're not going to be able to do it in isolation for a very long period of time. But we will get managers to that point. I'm a conservation manager myself. I started in this genomic space just 10 years ago. And so it is possible to get people to that point, but the problem we've got now is sequencing technology is going so far, so fast, and the bioinformatics with it. The gap is just getting ever wider. And so that's the bit we need to try and figure out how we're going to start bridging that gap. Because if we wait until Earth Biogenome has produced all the genomes, then you've got to take another 10 years after that. That's 20 years. That's a disaster in the conservation space.
Certainly. Thank you. Yes. Here's a question that a lot of people have responded to online. For those countries that have not used genomic data for conservation management, what is step number 1, 2, 3, 4, and 5 that you would recommend to begin to apply the same sorts of things that you're doing?
I think a lot of the responsibility comes back to the geneticists and genome biologists, that you know, I remember the first conversation we ever had with the Devil program about genetics, and one of the comments was, well, you know, there's nothing you can tell us that we can't already work out or have the information ourselves. And now we're so heavily integrated with that program. So it takes time and effort and communication and really partnerships and relationships to get people to the point where they can start using the information well. So that's where it starts. Like if you're working on a genome on a species, start looking into the ecology of that species and the threat status and what's happening with it, and start contacting the people that work with those animals on the ground and ask them what kinds of questions do they have and what kinds of information you know, do they need. And what you generally find is they have these questions and they don't even know that they can use genetics or genomics to answer those types of questions. And so that's where I would start.
So that's step number one, right?
Yeah, that's step one.
Get the manager.
And then there's about 100 after that, but that's a good place to start.
All right. Well, that particular person can contact you and maybe get a bit more of an outline if they want to,
Sure.
or read your book. That would be even better. A few questions about Tasmanian devil. The question here, is there any evidence that higher level heterogeneity could affect the fitness of the devils to combat this facial tumor cancer? You showed two distinct populations on either side of Tasmania. I mean, was there less? Well, you know what I'm talking about.
Yeah. So we know that the devils on the East Coast have different MHC diversity, which is MHC genes are particularly important in devil facial tumour disease, and Wan's presentation coming in this session hopefully will provide some more information around that. But and to the West Coast. So the disease has progressed a lot slower across the West Coast population, but it's still progressing. So there's no kind of resistance allele that stops devils getting devil facial tumour disease. What we do know is devils have inherently low genetic diversity. They've suffered a number of population bottlenecks over geological time. And so the hope is that by introducing new alleles into the system, we are giving the devils more adaptive potential than they currently have to hopefully get to the point where they adapt to live with the disease that they've got, because the disease, you can't eradicate the disease, it's not leaving the environment. And so what we do know is the populations, because of the fragmentation and the low genetic diversity that they have, they're actually more likely to succumb to inbreeding depression moving forward than they are to going extinct from the disease. And that work's being done by Catherine Gruber, who works with us at Sydney Uni as well. Yeah,
there was a related question about that are the more genetically diverse individuals, do they have higher reproductive success when you track them in captive populations?
No, funnily, well, they've got roughly about the same reproductive success. That's the work that we're still doing at the moment. So we only started doing the mixing releases in 2016. And so we're just monitoring those populations at the moment. So that's a watch this space in regards to the long-term fitness benefits of the F2 generation.
Okay. And one more question about the devils. Obviously, you touched a lot of people. And some people said they're so cute and other things. But here's a question. How did you quantify functional diversity in the devils? Was that based on SNPs, or was that based on overall heterozygosity, or what?
So that data that we presented was the functional diversity at the MHC region. Using the micro-linked microsatellites. One's just developed a new MHC typing assay, and we're also using target gene capture that we've developed using a MyBates protocol, and that target capture was developed from the reference genome that was produced in 2012 by Liz Murchison. So,
Here's one just about kind of getting back to that first question on what are the first and second steps of getting this going. Give us some nitty-gritty on how this interaction between the managers and the genomicists actually works. Is that a weekly thing that goes back and forth? Is it a monthly, a yearly? Give us some idea of how that works.
If we're leading up to a translocation, I would say it's every 2 hours. I seem to be on the phone. Yeah, it depends on what time of the year and what type of activities that we're undertaking. We definitely have a catch up with the management team every few months outside of the field season and any kind of definite conservation actions. If something exciting happens in the lab or we're finding some cool results, we tend to get on the phone quite quickly and start having a conversation with them and what we think those results may mean. So we do provide feedback on the project, you know, I would say generally every month across time, and make sure as well that we not only take the time to say what the results are, but kind of distill it down into language that makes sense to the conservation managers. The one thing with geneticists is we love our jargon, and we definitely love an acronym. So we make sure that we spend enough time explaining to the managers exactly what we mean and what we think the consequences of those results are.
I think participants really appreciated your portal that's developing, and probably everybody's waiting for it to be released. And one question comes along with that is, where do you get the funding for the software engineers to develop these sorts of things?
I'm hoping that in 20 days, I'm going to get a big fat grant that's going to allow me to pay for my cloud architects and software engineers. So at the moment, that portal that I showed in the presentation is just a mock-up that's been undertaken by Paris Brandes within our research group, who's very clever with software design and a very talented bioinformatician. So yeah, we work in close partnership with a cloud architect company down here in Australia, and Amazon Web Services. So our hope is that with Ronin and AWS, we can get it up and running live. But we're definitely looking for some funding support at the moment for some software developers.
Okay. I see another question comes up about the actual interactions between the managers and the genomicists. And the question is, what is the size of a team that could effectively carry out these sorts of transferring or translocations and transferring the information from the genomicists to the managers?
Well, when we started, I was the manager and Kathy Belov was the geneticist, so it was a team of 2.
Okay.
And it's definitely grown from there to I think there's about 10 guys down at the Devil program now that we work really closely with, and now we have a team of 10 or 12 depending on what's happening with the students and postdocs in our research group.
Okay. And here's a note or a question from one of our tropical participants. How do we encourage, or how would you suggest that we encourage governments that know nothing about conservation genomics to actually, you know, be aware of this and also begin to support this sort of technology?
I think, again, if you can find a poster species like we did with devils, so, you know, devils had a really key conservation problem. We already had a reference genome for that species in 2012 when we commenced the Tools and Tech project. And then just go and ask them, you know, what is their top 5 management questions that they need to get resolution on in order to be able to feel like they're going to move the recovery of the species forward. And in that conversation, you know, they'll tell you a few things and you'll say, well, you know, we can help with that because we can answer that question with genomics. And they'll go, what do you mean? And so this is what we did with the translocation. One of their key things is they wanted to know whether or not the devils that they were releasing were actually surviving and going on to breed. And you can invest hundreds of thousands of dollars in satellite collars and satellite tracking over a long period of time, or you can sample the animals that you've released, sample the incumbent population, and as long as there's genetic differentiation, you can sample the offspring to see whether or not you've had mixing at the crudest level in the population. And that's what we did. And so once they see the power of genetics in answering one question, then you've got buy-in. And as soon as you've got buy-in, you can move the project forward quite quickly.
Does it help to have charismatic organisms such as you have there? As a number of people here said, they're so cute.
Well, they are kind of cute until they bite you. Yeah, so it does help to have charismatic fauna. I'm very fortunate in working with the charismatic species, but, you know, some of the team, one of the members of our steering committee, Maurizio Rosetto here in Australia, he's a botanist and he works on plants. And I think it's about how enthusiastic you are about the species you work on and the work that you do. Is the more enthusiasm you bring to the conversation, the better buy-in you get from the other end.
Sure, Sure. There are a couple questions here about, and we're going to have to wrap this up soon, about education. And Eric Jarvis brings up the point that to fill this gap between genome and conservation biologists, we need experts in both fields on student committees. And so we need to begin to broaden out how we're training students to make sure that they appreciate this. Do you have any comments about that?
Definitely. So my guys will tell you, I'm always telling them that, you know, everyone who's participating in PhD programs at the university will get a PhD. My aim is to teach them how they can take that PhD out to the bigger world past academia and how to communicate their science. So we, as a research group, spend a lot of time with our postdocs and PhD students. If we're going to a conservation managers meeting, I try and take, you know, 1 or 2 of our team members with us so they can participate in the conversation. They can see what it takes to kind of distill our science down into a format such as the managers can understand. And yeah, just try and help upskill and train them during the PhD program. So once they've completed their degree and they go out themselves as scientists, it helps improve the communication.
Okay. All right, I think it's time to bring this to a close so people have a few minutes between sessions here. I just wanted to read one last comment from a passionate scientist that said, "Amazing presentation. I almost teared up. Keep up the excellent work. It always brings a smile to the face hearing about projects like yours." Thanks very much, Carol. Excellent presentation.